Just amazing the Wickenburg School District does not get it. Of course, if it was not for the smart average parents for the students in the Wickenburg School District, the school district would be like a reservation school. Because the parents in Wickenburg are excellent mentors for their children, and they will make sure their children make the grades and especially do the school work. That is the reason why Wickenburg is a unique environment compared to many communities across the United States. When Mr. Mathis was teaching biology classes on the Navajo reservation, he used such method to teach vocabulary terms to his students, and it worked so well. Also, when he taught science classes at Globe High School, especially the anatomy class, he used such method. His students loved it. The anatomy students loved learning how to pronounce the words correctly. Also at the Hopi High School he used such teaching method. The parents loved his teaching style. In all the school districts Mr. Mathis taught at, he had excellent support from his peers and especially with the school administrators. However, not at Wickenburg. It is so puzzling with the leadership in the Wickenburg School District, and especially with the human resource person. It is common knowledge on the reservations the students do not have excellent mentors like it is in the Wickenburg environment. The teachers in the Wickenburg School District are so lucky. There are several parents and grandparents who are highly educated. Without such support, most students in the Wickenburg School District could not produce excellent students. That is the reason Wickenburg is an excellent, unique environment. And it is not because the administrators and the teachers are doing an excellent teaching job. However, no doubt the teachers in the Wickenburg School District are forced to teach to the tests. Of course, the teachers in the Wickenburg School District are first-class individuals. Coming up is an excellent example how Mr. Mathis taught science vocabulary terms to his students. No doubt other teachers, and especially in the Wickenburg School District, the teachers should be developing the same method. However, what is holding the teachers back? Because the school district administrators are forcing the teachers to teach to the test, which is not teaching. The reason for such attitude is because more money for the school district through the government. This is no surprise at all for the Wickenburg school superintendent.
Here are your science words for today.
Nucleotide.
Macromolecule.
Deoxyribonucleic acid.
Gamete. Gametangium.
Nucleotide, any of a group of molecules that, when linked together, form the building blocks of DNA or RNA.
There are two types of nucleotides, purines and pyrimidines. Further, the two types of purines are adenine and guanine. Also, the three pyrimidines are cytosine, uracil, and thymine. Now, to remember the purines, the first mnemonic is pure as gold, where PUR in pure refers to purines, A for adenine, and G in gold for guanine. The second mnemonic is pure silver, and chemical formula of silver is Ag. Again, PUR in pure refers to purines, A and G for adenine and guanine. Now, you also know that purines have two rings in their structure. To remember this, just imagine that you have two gold or silver rings on your fingers. Moving on to pyrimidines, the mnemonic is cut the pie, where C for cytosine, U for uracil, and T for thymine. And pi in the mnemonic will help you to remember that these are pyrimidines. You also know that pyrimidines have one ring in their structure. To remember this, the trick is that pi means one circle or a ring. Now, let's move to pairing of the nucleotides in DNA and RNA. In DNA, Adenine pairs with thymine and guanine pairs with cytosine. Just remember ATGC. This implies that A pairs with T and G pairs with C. Please note that RNA has uracil in place of thymine, so adenine pairs with uracil in RNA. You also need to remember that A, T and AU have 2 hydrogen bonds, whereas G and C bond has 3 hydrogen bonds. We have a mnemonic for this as well. 2 bonds, which implies thymine in DNA and uracil in RNA pairs with adenine with 2 hydrogen bonds.
Macromolecule, a very large molecule, as a colloidal particle, protein, or especially a polymer, composed of hundreds or thousands of atoms.
Today we're going to talk about macromolecules. Macromolecules are large structures that make up all living organisms. All macromolecules have small subunits called monomers, which bind together to form larger units called polymers. And there's 4 classes of macromolecules. They have carbohydrates, lipids, proteins, and amino acids. And I'll talk about each of these in turn, starting with carbohydrates. Carbohydrates are the main fuel source for most living things, as well as a structural component for many plants. Monomers for carbohydrates are called monosaccharides. Saccharide means sugar, so monosaccharides, one sugar. These are simple sugars like glucose, fructose, and ribose. Then your polymers are called polysaccharides, and they are long chains of monomers or monosaccharides. There's storage carbohydrates like starch in plants and glycogen in animals for energy storage, and then structural like cellulose in plants and chitin in insects. Moving on to lipids. Lipids have a variety of functions in living organisms, and there's a lot of things lumped into the category of lipids because they're all hydrophobic. All right, some functions include energy storage, cell membrane formation, and steroid function. They have no real monomers. They're just they just are what they are. So fats include triglycerides and saturated and unsaturated fats. You also have phospholipids, which form the cellular membranes, and then steroids, which include all sex hormones, and all the steroids form from a cholesterol base, and all those are included in lipids, so it's pretty diverse. Then you have proteins. Proteins are involved in nearly all cellular functions and are a major part of all living organisms. Their monomers are called amino acids, and there are 20 different amino acids. And their polymers are called polypeptides. You bind amino acids together to get polypeptides. And the protein structure results from folding, which determines the function. And lastly, you have nucleic acids. Nucleic acids store, transmit, and express genetic information. So their monomers are called nucleotides, like adenine, guanine, and thymine. You may be familiar with these terms from studying DNA and RNA, and that's what their polymers are. They're called polynucleotides, and they're strands of DNA and RNA.
Deoxyribonucleic acid, an extremely long macromolecule that is the main component of chromosomes and is the material that transfers genetic characteristics in all life forms.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which 2 strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT or TA and GC or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.
Gamete, a mature sexual reproductive cell, as a sperm or egg, that unites with another cell to form a new organism.
The two gametes of sexual reproduction are sperm and egg. Did you notice the large size difference between the sperm and the egg? There's a reason for this, but let's see how their formation differs from one another first. The creation of sperm is called spermatogenesis. It starts with a diploid cell called a primary spermatocyte, and after meiosis I, it forms two secondary spermatocytes, which are haploid, but have a duplicate chromatid with them. After meiosis II, all the cells are haploid, which in humans means they'll have 23 chromosomes. The haploid spermatids will mature into sperm, which will then be motile and ready to fertilize an egg. The creation of an egg, also known as an ovum, is called oogenesis. Like spermatogenesis, will start with a diploid cell called a primary oocyte. But after meiosis I, the cytoplasm is divided unevenly, and one cell gets most of the cytoplasm. The cell with the most cytoplasm is called the secondary oocyte, and the other small cell is a polar body. Both of the cells are haploid, with all chromosomes duplicated. After meiosis II, the secondary oocyte is again divided unevenly, and most of the cytoplasm goes to the ootid, which will mature into an ovum. This ovum will have all the organelles, including the mitochondria. Even though only one mature ovum is created by the one oocyte in oogenesis, females are still born with millions of cells and will release hundreds in their lifetime. Not every cell created by one individual is the same. In fact, they're all very different. Genetic recombination from crossing over and independent assortment during meiosis lead to genetic variation and differences in each sperm and egg. This is why brothers and sisters don't all look identical. Now, the whole point of making sperm and egg is for fertilization, to make offspring, babies. Once the haploid set of chromosomes from the sperm are released into the egg, the two sets of chromosomes combine, creating one diploid zygote, which will grow and mature over time into the offspring. We all started out this way, just a tiny cell the size of a grain of sand. But sometimes things don't always go so smoothly. Errors during meiosis can cause non-disjunction. Non-disjunction is a failure of homologous chromosomes or sister chromatids to separate during meiosis. Here's one of those cases. During metaphase I, everything is lined up as expected. But during anaphase I, a chromosome is pulled to the wrong side, which leads to gametes with incorrect numbers. Some will have an extra chromosome, and some will have one less. This could lead to a variety of issues. While most of these cases will lead to miscarriage, some disorders are not life-threatening. Here are some examples of disorders that can occur as a result of non-disjunction. Probably the most widely known one is Down syndrome. In this case, there is an extra 21st chromosome, which can lead to mental impairment and stunted growth, among other things. Klinefelter syndrome occurs when there's an extra X chromosome in the standard male karyotype. This can lead to weaker muscles and bones, less testosterone, and sterility. One of the only cases of monosomy, where there's one less chromosome, is Turner syndrome. In this case, the second X sex chromosome is missing. The female will be sterile and have shorter stature and a webbed neck, but may not have any mental impairments.
Gametangium, an organ or body bearing gametes, as in mosses and liverworts. Yes, mentorship is the best way to teach. And I bet you $10 the students who are making excellent good grades at Wickenburg High School, they have excellent mentors in their home environment. One thing for sure, students these days should have no problem developing an excellent vocabulary, but only if teachers are being more innovative with technology for their students.